Hey everybody, Mr. Cars again. I'm just going to give you another video here to try and help you with investigation 6.2. It's all in the Strand. This investigation actually has two parts. The first part should be fairly simple. The second part can get a little tricky, so you want to make sure that you read all of the instructions and please pay attention, rewatch the video as many times as you need to. So if you remember from the DNA model activity that you should have done earlier this week, that mod gave you the basic structure of DNA. DNA is a ladder shape where the sides of the ladder are made up of alternating sugars and phosphates, and the rungs were made up of those complementary nitrogen bases. In this activity, we are going to use the same concept, but we are only going to be looking at one side of the ladder, and instead of actually using the names sugar, phosphate, and either adenine, thymine, guanine, or cytosine, we're just going to use these colored dots that you see here. Each dot represents an entire nucleotide, so one sugar, phosphate, and base, and the color of the dot will represent which base we're using. Now, we're not going to worry, again, like I said, about the names or anything, but just understand that because those colors represent the bases, they will be complementary, meaning they will only pair up with the alternate color. And those complementary pairings can be seen here in table 6.1. All right, so wherever you see a red dot in DNA, that will correspond to a blue dot in RNA. A blue dot in DNA will correspond to a red dot in RNA. A green dot will correspond to a yellow dot, and you don't see it here in the chart, but if you see a purple dot in your DNA, that would correspond to a green dot in RNA. All right, so in order to do this lab, we have to understand what we are trying to do. So here we are trying to find out how we can read the message, the code, if you will, that is in DNA. And in order to do that, the DNA code has to be brought from the nucleus of the cell where the DNA is into the cytoplasm where it can be translated or decoded by a ribosome. DNA itself cannot leave the nucleus. That would kill the cell, which of course would not be a very good thing to have happen. So in order to get the code of DNA out of the nucleus, we have to transcribe it first into a molecule known as RNA. And RNA is just a complementary copy of DNA that can be taken from the nucleus and read or translated at a ribosome. So in order to make RNA in the first part of this activity, you are just going to look at a strand of DNA and you are going to make the complementary strand of RNA according to this table 6.1. So when I have a red dot in my DNA, that means I'm going to place a blue dot for RNA. A green dot would place a yellow dot, a blue dot would place a red dot, and so on down the line. All right, so you'll keep on referring back to this table 6.1 until you are comfortable knowing that red goes with blue and blue with red and so on down the line. When you are ready, you will come to this slide. All right, this is the DNA strand that we would find in the nucleus of our cell that needs to be copied into RNA so that it can get out of the nucleus and be translated. So to do this, I will start with green, my first color in my DNA strand, and I know from coming back to my table that green is complementary to yellow. So I will start by coming down here in the bottom, grabbing a yellow dot and dragging it up top. I'll go back to my table and see my second dot is red, which means I need to grab a blue dot, which is complementary to red, and put it in my strand. My next dot is purple. Purple was complementary to green. And then I'll have my first three dots of my RNA strand done. Now that's not very difficult. All right, you're just basically following the table here and grabbing the alternate color for each of the dots in my DNA strand. The part that can get tricky, and it happens to me if I try to do this whole thing every time, is that I'll end up getting here somewhere in the middle and I'll lose my place. So then I have to go and count how many dots have I done, which can just become monotonous and annoying, you know, to do. So what I typically do is I go 3 at a time. Reading DNA 3 at a time, that's how you're going to do it when we go to translate it. So I just stick with the 3 at a time. You can do it 1 at a time, 4 at a time, 5 at a time, however you want. But when you're ready to kind of, you know, cross some off, you come up here into the top, into these tools area, and you'll see this line feature. All right, and you can just kind of draw a line I did my first 3, so I draw a line down there. That means I know I did those 3, and I could just move on to my next 3. All right, if you want to draw lines every 3 and just break it up like that, you can. However you want to do it, that makes it easier for you. So for my next 3, I know I have a red dot. Red was corresponding to blue according to my table, a blue, which corresponds to red, and a purple, which corresponded to green. Again, I know all of that from coming back here. DNA dots are red, goes to RNA dot is blue, and so on down the line. I've done my next 3. I'd come up here. I'd make another line. I know I have my first 2 sets of 3 complete. And I'll just continue on down my DNA strand, adding the complementary dots to RNA until I have a strand of RNA that has the same number of dots. I think there's 36 in DNA. Once I have that done, I am now ready to decode that message. All right, and the way you decode a message is through a process known as translation. All right, translation is done in a ribosome in the cytoplasm of every cell. All right, and in order to translate it, you are going to read the sequence of dots 3 at a time. All right, and each 3 dots is going to code for 1 amino acid. And when I link together several amino acids, I will build a protein. And of course, proteins are what control most of the processes in our body, and proteins are what determine all of our traits. So here, I'm going to be referring back to this table 6.2. This is kind of the decoding table, if you will, that we will be using. So here I see table figure 6.5 again, where I took my DNA strand and I coded it into RNA as an example. So I am going to read this 3 bases at a time. So I look at my 3 bases and I have blue, yellow, red, blue, yellow, red. So it's the same 3. When I know that, it's going to end up being the same amino acid for both. So to do it, I come over here. I see first bead in table 6.2. Well, my first bead is blue. So I'm going to be here in this blue section. So somewhere between these 2 lines that blue makes, I'm going to be in there, kind of narrowing it down to 16 different colors that it can be. My second bead was yellow, or my second color was yellow. I'm going to come over here and I've now narrowed it down to 4. It can be blue, blue, yellow, yellow. My third is red. That's going to be the one that tells me which color it's going to be. I see red is right here. So I'm going to be right here, and my first chain is going to be blue. All right, and that is how that will work. All right, now, when you're ready to do it, there's some further instruction on how to do it here. First bead, which will narrow you down into this section. Second bead was yellow, which brings you down to one of these 4. And then that third bead being red is going to be the one that tells you which color it's going to be. When you are ready, I have placed figure 6.2 back here. Again, you are going to have to kind of keep flipping back so you can go 3 at a time and read your RNA strand. You may want to draw lines again in your RNA strand as you code it so that you know which one you are going to be doing so that you can get through there. So if I do it, I know that my first 3 were yellow, blue, green. So I will come down here. Here's yellow, so I'm somewhere in this area. Second bead is blue, so I'm going to be in one of these 4. And my third was green, so I'm going to come over here and see that where yellow, blue, and green connect, that gives me a yellow C in my protein chain. So I'll drag a yellow C up there. I'll come back over here and I'll see my next 3 are blue, red, green. So I'll be blue in the red, so I'm right here in one of these 4. Green is going to put me right here. So my second dot, or second C, or amino acid in the chain, is going to be blue. I will then go back and do my next 3 and keep on coming back, and eventually I will chain a bunch of these C's together in a certain colored pattern. That chain of C's, that chain of amino acids, would make a protein. In the color of each of those chains, the order of those amino acids will determine which protein is made. Again, that second part can be tricky. Watch the video again if you need to see how I did it. Use this table here to kind of see. This would be your 3 dots that you're looking at. All right, as you go to the first dot, second dot, and then third dot, how you get that color of amino acid that you need to put in your protein chain. If you have any questions, you guys, again, have been doing a great job at asking me through private message in Google Classroom or through email. I will respond to either way. Please keep up the good work. Stay healthy. Stay safe. Hope to see you soon. Bye.